Hola a todos, bienvenidos al segundo día del Festival Suratómica. Estamos muy contentos de dar inicio a este día con una charla increíble que nos va a compartir el artista Paul Vanouse. Esta charla va a ocurrir en inglés, entonces, ya habiéndoles dado la bienvenida, me paso a presentar a Paul y me paso entonces al inglés. Welcome everyone. Thank you for joining us today in the second day of the Suratómica Festival. Today we have a great lineup of events, and to kick it all off, we have an amazing guest, Paul Vanouse, who's gonna share his amazing work with us today, the frontier between art and science, which is an overarching theme of our festival. Paul is the program head of the emerging practices at the University of Buffalo. His work is deeply interdisciplinary and very much about about amateurism as perhaps a way of conceiving work. And he has a very broad spectrum of pieces and practices that all formulate a fascinating vision of what art and science do and how they relate with each other. I'm gonna give Paul the chance to tell us about his work. Can't wait to hear more. Welcome, Paul.
Thank you, Simone. First of all, I guess I should thank Simone and Juan and Daniela and Natalia and others for inviting me for this. I have to say I'm very happy to be part of the festival. When we first started planning this, though, I was really looking forward to being physically present, really engaging in the workshops, and particularly this thing of the dialogue that's been created between Suratómica and CERN and Switzerland. I was particularly excited by it because it seemed to make the world smaller and connect initiatives that I believed in. And it shrunk these kind of national and disciplinary and economic boundaries. So it's in the midst of the fallout, political and pandemic fallout. These kind of opportunities, I guess, are more complicated. But I guess we're here now, so I wanted to make the most of this moment. Opportunities to converse. I have the meat background here, which is part of a project called the Visible Human Project of 2015. Really my introduction, the first biology-based piece I made, and it's kind of a segue into the panel that's going to happen later with Simone. But for now, I think I'll try to share my screen.
Yes, please.
And hopefully this shared screen just shows slides and not notes and not too much other stuff on the screen. Good.
Yes.
Okay.
Just the images. Perfect.
Okay. Great. Let me then start. Okay. So Simone mentioned I'm an artist who works in emerging technological forms. My work addresses complex issues raised by emerging technosciences self-reflexively, using the very technosciences as media for artistic explorations. For nearly 20 years, I've worked with DNA as a medium and molecular biology protocols as artistic techniques. Radical interdisciplinarity and impassioned amateurism have guided this work. And in this talk, I'm hoping to present not just the work, but also strategies I've taken to create meaningful interdisciplinary and often sort of challenge reductional ideas in the technosciences. The goals have always been enhanced participation and positive transformational dialogues across the disciplines. I I thought, let me go back a tiny bit. I thought I'd begin by showing not just the work, but I also thought I'd show some of the artists that have inspired me, particularly in the 1990s. Then I thought I'd go into my works with biology and DNA. And then finally, I thought I'd show you a slide of my endeavor here, the Coalesce Center for Biological Art here at Buffalo. The work that excited me most as I was beginning my I guess what you call my adult work were those that were not really discussed in any of the studio art programs of the time. They were these postmodern works that seemingly broke all the rules that I'd studied as a student. The most influential artist for me in this was Jenny Holtzer, whose work I first saw the year I graduated college in 1990. And what fascinated me was the way that her work would appropriate the media of advertising to reflect on mechanisms of power and the manufacture of desire. This piece here utilizes this spectacular LED medium through which we're typically seduced by advertisements and embeds it with this critical reappraisal in the very heart of capitalism, Times Square. This was a way I very much wanted to participate in framing meaning and culture. Other work from the period included Mining the Museum by Fred Wilson. And in this work, Wilson recontextualized objects from the museum's permanent collection, and in so doing radically alters how viewers make meaning. The denigrating sculptures, typically based on fanciful illustrated representations posed outside cigar shops in the 19th century, turn their backs in this case to the gawking museum audiences. Instead, they gaze firmly upon photographs of Native Americans taken at the time. The audience member in this case is not placated, but ignored. Simply by turning the statues around, they become disobedient subjects of their own desires. My work is conceptual and influenced heavily by postmodern art practices such as these, but I'm also deeply engaged in the material aspects of my media. My laboratory work deeply informs my theorization. In my work, I'm continually questioning ideas of difference, how it's constructed. How are we differentiated into races and types? And how are we differentiated from one another? Part of this fascination probably comes from my own experiences as an artist of Afro-Caribbean descent. I've been categorized as Black, white, brown, mixed, biracial, Latino, other. And what I'm particularly focused in is how these technosciences are complicit in this. But I don't mean to say this is just a scientific process, but it's rather a really complex social process. And so Bruno Latour has described that if one wants to dispute a fact or an assumption, it's necessary to reopen the black box, to build a counter laboratory. In much of this work, I take this method extremely literally, to literally counter entrenched cultural constructs. The work I'm going to show is all of it really stemmed out of an invitation to a roundtable at the University of Washington in Seattle in 1999. It was a roundtable with some of the most important genome scientists to discuss the implications of the Human Genome Project. The goal of the Human Genome Project, as you probably mostly know, is to map every gene in human DNA. There are about 30,000, and approximately all the base pairs, which is there are about 3 billion. And there was a great deal of hype around this project. For me, some of the most problematic hype were all the misleading metaphors surrounding DNA. The idea of DNA as destiny, the code of codes, and particularly this metaphor, DNA fingerprinting. The metaphors are problematic for a few reasons. One, their fatalistic determinism. to the emphasis they place on DNA as a code at the expense of a more nuanced understanding of the material, the organism, and the environment. And 3, the exaggerated belief that such metaphors create in forensic and identity fixing technologies. So in all my work, I've tried to complicate fixity and wholeness and essence, determinism, in favor of dynamic change, hybridity, multiplicity, and uncertainty. And at the heart of this has been investigating claims of difference and of sameness. Key to all these investigations has been to understand scientific images as cultural images, and to trace the social processes through which these came to be accepted. This is very much like the tools of visual studies. So here in this way of looking, I'm trying to utilize all the tools of being a postmodern artist. All the projects I'm using, at least in the beginning, use this analytical tool called gel electrophoresis as an expressive medium. Just like Jenny Holtzer appropriated the LEDs, I appropriated this technology as something I could critically reinvest. The images created with this technique are often referred to as DNA fingerprints. And in my work, I'm always emphasizing this sort of malleability of this medium. Here's how it works. Again, I hope this is understandable given that I'm so far away and I can't demonstrate with my hands and such so much. The way it works is that in slide A, you can see you chop DNA into a few, into various fragments. So this, in slide A, you can imagine that you started out with 4 pieces of DNA and you chop them into parts. Some basically into 2, chop them almost in half, but one is a larger fragment and the other is a smaller fragment, and that there are 4 of them, but now there are 8 fragments, 4 large and 4 smaller. In B, you can see that you can imagine if I inserted those fragments into a small slot in a piece of gelatin and applied electricity across that gelatin, the DNA would be pulled through that gelatin at different rates. The reason for the different rates is that, as you can see in slide C, the small little pieces move faster than the big ones. That's all there is to it. And then that's the basis for the DNA fingerprint. It's really just a method of kind of showing visually the different sizes of DNA in a sample. So following the rough draft, let's look at the year 2000 when this work of mine really began in earnest. In 2000, there was this statement that came out in a lot of the press that said there's no scientific basis for race. And this came out from a team of UNESCO scientists following the rough draft of the Human Genome Project. The issue was a concern because the Human Genome Project marked a return to interest in human genetics not seen since eugenics. The original title for the Human Genome Project was, in fact, neo-eugenics. And it was the Nobel laureate James Watson who suggested a name change in the 1980s. However, as optimistic as it was that this statement was made, there's no scientific basis for race, I assumed that race could exist even, or that racism could exist even if race didn't. That it would simply leave the body's surface and go molecular. That now, rather than declare the racially stigmatized body as genetically unfit, we would describe certain genes common in stigmatized populations as unfit. Molecular racism is the term for this. And I recalled that the book Race Crossing in Jamaica, written by the American eugenicist C.B. Davenport in 1929, described Black-white racial hybrids as less than the sum of their parts. This struck a chord for me because my biological family members are racial hybrids of Jamaican descent. So in a sort of historical condensation of eugenics and genomics and identity, this first project, I wanted to construct a dramatic experimental system to finally determine who in the family, which skin color is the most genetically fit, quote unquote. quote. And how better to determine this than first extracting individual skin color genes from every family member, which in this case involved inviting a doctor friend over to dinner, as well as my parents and my sister, shown here, and extracting blood. The experiment used the current medium of genomics, DNA fingerprinting, as my medium, but I altered the way it was interpreted. So I inserted the family skin color genes into unique lanes of a giant DNA gel to see which ones would move the fastest. Kind of ironically thinking, wouldn't this be an appropriate method to determine our fitness? Again, you've seen how this technique works. So, you know, the larger fragments are going to move slower than the smaller ones. In this piece, then, the skin color genes can be raced in an experiment that philosopher Bill Eggington described as a race about race, in which the actual bodies were now erased. One of the things I was also trying to demonstrate with this, in terms as a true counter lab, was I was trying to show that race is in fact a verb, that subjects can be actively raced or racialized. We are not born into a race. It's a machinic operation. And now this operation is an assembly process that draws on a whole network of sources and forces, everything from scientific literature to poetic metaphor to state policies, national boundaries, economic structures, clever puns. The instruction device here then creates visual evidence. It's a complicated machine that literally racializes. Here's a little bit about how it, if you're actually watching the installation on the back screen, if you're actually watching the installation on the back screen, you would see this this process unfold where the DNA's position in the gelatin is analyzed by computer. This is like a machine vision system I built that would keep track of which sample was in the lead. I still show this piece even after 20 years because I still kind of can't believe that it was possible to build at that time. It was kind of took everything I had. But on the other hand, it was an extremely pessimistic view that new technologies would be used to reinforce familiar stereotypes. But shortly, just as I was finishing this piece, James Watson, the Nobel laureate, who famous for the discovery of the structure of DNA in 1952, which was problematic for its own reasons. But anyways, this Nobel laureate gave this talk at UC Berkeley. In the talk, he described an experiment by colleagues in Arizona, and the colleagues injected patients with an extract of melanin. They intended to test whether they could chemically darken the men's skin as a form of cancer protection, only, as Watson said, to observe an unusual side effect, which the men had developed these sustained and unprovoked erections. This is in James Watson's words. He said that this melanin injection is even better than Viagra because you don't even have to think about sex. That's why I have Latin lovers, he said. You've never heard of an English lover, only an English patient. There's something rather comic about this lecture he gave, but on the other hand, it's also something very dark, and that is that you have a Nobel laureate here showing us now that that racism has left the skin, right? That it is now no longer Black people who cannot control their libido, but rather this molecule melanin or even Blackness itself, which is to blame. Indeed, perhaps it's not the Black body that's prone to promiscuity, but Blackness itself. When I finished that work, I realized that it was appropriate to really look into this metaphor of the DNA fingerprint. The DNA fingerprint had become the most authoritative cultural image, right? The very difference between being guilty and being innocent. But what is a DNA fingerprint anyways? And why is it sometimes called so? If it doesn't come from a finger, and it's not a print, it's a metaphor to an earlier biometric technology. I showed in the last piece that it doesn't come from a finger, it comes from blood samples usually. And it certainly isn't a print, it's a mobile process, the electrophoresis image. It's a metaphor and a curious history that the metaphor brings along with it. So DNA fingerprinting, is obviously this comparison to real fingerprinting from an actual finger. That history, the fingerprint was first used in colonial India by Sir William Herschel, originally to prevent fraud on contracts and deeds, but he later proposed, he later fingerprinted prisoners on sentencing and proposed a much broader use of fingerprinting on the whole population. To get a sense of the atmosphere of colonial India at the time, in 1871, the quote-unquote Criminal Tribes Act was passed, which mapped onto the existing caste system policies to deepen notions of race, class, and criminality as linked, inherited, and predetermined. Fingerprinting then is further explored by Francis Galton, the cousin of Darwin, father of eugenics. In the 1890s, he thought this might finally be a way of discerning the elusive physical markers of heredity, ethnicity, and race, as well as to establish individual identity. Much like DNA is today, fingerprinting was seen as a way of unlocking the keys to nature's secrets, hidden secrets of human identity and propensity and background. To Galton's dismay, however, fingerprints didn't show one's history. It was actually pretty good in terms of differentiating people, but not in terms of grouping people. So jumping back then to the present, law enforcement loves the metaphor of DNA fingerprinting. And I think this is a really strange quote. We like the term DNA fingerprinting because it invokes in the mind of a jury that we're identifying one individual to the exclusion of all others. What was so strange about it was to hear a law enforcement person describing the reason for liking something was what it invoked. It was almost like they loved the pure poetry of it, not the science necessarily, right? Just what it meant to a jury to say the word fingerprint. This metaphor is explained by the inventor of the technology, a guy named Alec Jeffreys, an Englishman, who writes, if we had called this something different, like idiosyncratic Southern blot profiling, what it was, nobody would have taken the blind bit of notice, but call it DNA fingerprinting and the penny dropped, right? Everything was in the name. Which brings us again back to one of the central kind of theorists, I guess I'm discussing in this talk, Bruno Latour. This is a cartoon from his Science in Action book from about 20 years ago. Latour shows two faces of science. One, the old face, which is one that we kind of come to understand from textbooks, which is once the machine works, the people will be convinced. But then this younger face of science says, no, the machine will work when all the relevant people are convinced. Right? So again, this kind of showing the kind of ways in which cultural proofs are the kind of basis of what works, what is factual. So in the next piece I'll show you, called the Latent Figure Protocol, I wanted to take advantage of all the different ways that we're of making a DNA image. This image shows a whole collection of all the different kind of chemical enzymes one can use to chop up DNA into those pieces. There are about 100 different ones. Each one cuts at a different base pair location, and they can be combined together to make literally hundreds of thousands of different permutations or patterns. So for instance, remember that shot we saw before of how DNA can be cut. This is what's called a genetic map of a plasmid. The two enzymes in pink show the base pair locations at which, if you added these enzymes to a solution of DNA, this is where they would cut that DNA into a big piece and a small piece. That's how it works. And then, as we remember that diagram from before, if you have big pieces and small pieces and you squirt them into a hole of a gelatin and electrify the gelatin, the small pieces will move the fastest through that gel. From this phenomena, then, I realized it would be interesting to try to do molecular biology in reverse, right? Since it seemed like the DNA sequences of most organisms were known, and since each enzyme cuts at a particular DNA sequence, and since the relative speed of DNA could be formulated, I should be able to write a computer program in which I could insert a sort of ideal image and have the program generate then a best image to, basically a recipe to make that picture. And this was the actual software program I wrote. I wrote it, a funny program, Macromedia Director, in about 2006. Of course, I wasn't interested in making these pictures just on the computer. I was interested in making physical material-based DNA pictures. So it was important to me to actually perform the image making live. In this case, I don't wear white like a scientist. I wear black in my counter laboratory. I also wanted to kind of signify that, like an artist wearing black, I'm creating artistic representations. And I was literally trying to show that I was literally trying to kind of undercut the authority of the DNA image, to dispute the authoritarian omnipotence by wearing the black of an artist, somebody that which we know is a subject position, which is oftentimes not trusted. The images in this case would form over the course of about an hour, and then they'd suddenly dissolve over the period of about a week. I would make, sometimes I would fix the actual images that were made. These are just photographs of literally looking down at the gel. And what I would do is I would superimpose on the images the actual DNA recipes that I used to create this image. I felt like this was like a scientific proof that the DNA fingerprint is in fact cultural. Again, part of this kind of notion of the counter laboratory, to show that the DNA images constructed, I literally construct them into these incredible, these sort of controlled shapes. And I've done several related projects to that. One of them, which is, again, the epitome of a counter laboratory, is a piece called the Suspect Inversion Center. This was between 2011 and 2014. In this piece, the idea was to be even more specific, to try to reproduce in this exactness the DNA evidence from a historic trial. In this case, it was the O.J. Simpson trial of 1994. But the idea was to basically reproduce this DNA picture, but using only my own DNA. And there's something that the title is Suspect Inversion Center, which was itself an anagram, SIC, of the popular TV show CSI, right? I was trying to very much kind of undermine this notion that DNA fingerprinting is this quick process that occurs in this kind of cultural vacuum of the lab. In this case, it was also important to me. I performed this with artist and lawyer Carrie Sheehan, and we used only my own spit to extract all the DNA. And it was important to me that I also play all these different roles. The O.J. Simpson, a white woman, Nicole Brown Simpson, and another victim, Ron Goldman. One of the things that occurred to me when I made this was that it's interesting that I never even had to get my own DNA sequenced to be able to kind of have a certain kind of mastery of understanding about where it would cut and how it would move. And this is because I didn't need to get my own DNA sequence to do that. I didn't need to understand my exact DNA codes because most DNA is identical, right? Most of our DNA is the same. So I could use the regions of DNA that don't change, even though that like all these fingerprinting techniques are based upon these tiny little regions of our DNA that might be a little bit different. The vast majority of it's the same. And those are the regions that I was using to construct this image. So that became the basis for the final DNA imaging project I'll show you, a piece called the America Project, in which I create DNA pictures of power, in this case, the US flag and the European crown. The object in the center I call the biomatter anonymizer. Audience members empty cups of their own spit into the top, and using the massive centrifuge below, I extract what I describe as their promiscuously commingled DNA. Right. In this case, it's interesting. I couldn't individuate anybody who spits into this because there are hundreds of different people whose DNA and cells get all intermingled. I realized that since most of our DNA is identical, at least 99%, I can create completely stable images even with hundreds of different DNA samples all mixed together. The project here then was a culmination of the attempt to experimentally prove my artistic manifesto of radical sameness. Our DNA is mostly identical. Technologies have been suggested to underline all our differences, but the differences are incredibly slight. The majority of us are just the same. I'll show a little snippet of how these images unfold. With all of these DNA projects, I'm working against the idea that the DNA image is natural and that the DNA image is some kind of an imprint, when it's actually just a temporal process which is often stopped short. Here's a bit of how the image actually unfolds. In this case, it's the image that seemed the most loaded and ambivalent, time-sensitive of them all. An icon here of utopian promise, a kind of melting pot, yet simultaneously a space of xenophobic nationalism, infinite biopower. In this image that you're seeing form, it's sped up by about 100 times, but I love the fact that in exhibitions it runs so deliberately slow that it takes hours to unfold. In this case, the long stripes or smears that I've created were actually quite hard to produce with DNA separation technologies. It's typically what biologists try to avoid. So my collaborator on this, my scientific collaborator, Solon Morse, had fun reading the troubleshooting sections of DNA imaging manuals to get a sense of how to intentionally glitch this process, how to produce exactly what most people don't want. I'll show you some little snippets. This image is unfolding as we move a little further. We get into this. I often downplay the kind of aesthetics of these pieces and as I describe the concepts involved in them. But I mean, I'm also, this is kind of a remarkable phenomena to watch, these kind of like emerging blurs slowly kind of moving into place to make an image. It's almost like an algorithmic image, but in this case, it's an algorithmic image created live in real material. I also wanted to emphasize this kind of idea, this kind of icon of America as being also this certain kind of notion of a kind of process, right? That there's also this sort of notion that this kind of utopian ideal being some kind of a process-oriented process-oriented experiment in its own right. Anyway, so moving on past the DNA work, I say that the DNA work reacted against the idea of DNA as fully denoting who we are, what will be, our strengths, our weaknesses, our genetic destiny. But interestingly enough, since about 2007, human identity itself has become increasingly complicated in a refreshing way by research around the human microbiome. The research here finds that bacterial cells in and around the human body vastly outnumber human cells. Furthermore, skin bacteria of the lower arm have said to make up, been found to have about 70% variance in strains between individuals, meaning that there's more difference sort of between our DNA than, or in our bacterial microbiome than there is with our DNA. In this context, then, the idea of what makes up a person is again under the microscope, and not just for scientists, but for the culture at large. For years, we've been debating who gets to be considered a person and when. It's a question that dominates our political discourse of the last few centuries because of the connection between labor and liberation in the post-Renaissance world. Well, how do we define human beings now? And to further dethrone any kind of simple conception of self, hope there's a little bit of giggling about this image. It's meant to be a little bit of release for you all. Most people think the way that they smell is part of their own natural condition in a way, like your smell is your smell, or your smell is your smell. But it's far more complex than that. There's basically a cocktail of microorganisms that live on and in us, which are the culprits to determine this distinct human feature. Human sweat actually has no smell. It's simply the smell of these bacteria that feed upon our excretions that produce the familiar acrid and sour scents. And in this sense, then, the smell of exertion, labor, is not actually a human scent, unless we're willing to redefine what constitutes a human. And in this case, scholars like Elizabeth Gross and Rosie Bradati and Monica Baca have sought to recast humans in a more sort of structural role, which privileges metabolic processes and flows rather than the organism. In this sense, the human being then is more like a tube along which chemical and biological chemical reactions are facilitated. Elizabeth Gross points out that force precedes subjects, and that this needs to be understood in terms of its full subhuman and superhuman resonance as the inhuman, which both makes the human possible and at the same time positions the human within a world where force works in spite of and around the human, within and as the human. On a whole nother front, industrialization, since industrialization, the factory model has radically changed from human labor to machine labor, and increasingly in the 21st century to microbial manufacturing. Today, non-human life produces a whole range of products. Enzymes, foodstuffs, beverages, feedstocks, fuels, pharmaceuticals, and in many cases, genetically modified new species have been invented for the specific product they produce. In this case, bacteria literally live to work. These new industrial processes then also point to a deepening of the exploitation of life and living processes. The design, the engineering, the management, and commodification of life itself. So the intent of this next project, Labor, is to paradoxically produce the scent of human labor, not as an end product, like in the pharmacy, in the factory, but as a byproduct. Not as a end product, as in the factory, nor as a byproduct or waste. The idea was to produce the scent as an end product, as a kind of intentional output of this kind of factory, a generative living process, as an art installation that fills a gallery with the scent of human exertion or toil. I wanted to reflect this idea of the industrial shift from human and machine labor to this increasingly pervasive form of microbial manufacturing. And in this process, then to basically contemplate the changing borders of what is human. So I began with working with capturing my own skin bacteria. There are basically 3 species of bacteria that are responsible for the scent of human sweat: Staphylococcus epidermidis, Propionibacterium, and also Corynebacterium bacterium. And these 3 species produce these kind of funky, sour scents, and also these kind of athletic, sort of more salty bits, oniony scents, all these sorts. Again, I guess the emphasis of this piece was that these aren't produced by the human, but they're produced by the metabolism of the bacteria. Then, after I extracted these, subcultured my own body for bacteria, I harvest the individual colonies before they could be incubated at a larger scale in fermenting systems. I should add, this was part of my process. In the actual piece, I don't use bacteria from my own body. I use bacteria purchased from the American Tissue Type Collection. And part of this is so that exhibition venues know that there's nothing accidentally in there that isn't safe. What I did with this piece is I also had to concoct custom nutrient mixtures that contains the right amount of required fats and sugars while not having too much scent of their own. I wanted to make a system that didn't just smell like a petri dish, but literally like our underarms. So I realized in making the system that I could use a lot of salt, for instance, because the bacteria that lived on the human body could handle high degrees of salt. Then the final project here, the Labor project, then was an art installation that recreates the scent of people exerting themselves under stressful conditions without any people, right? There are no people making the smell. It's created by bacteria propagating in the 3 bioreactors, which you can see here, one on each side of the picture here, and then one in the back. Unfortunately, I can't produce the scent for you here, nor even the sound element of this piece. I really consider this not just to be a scent installation, but a multisensory installation. Each bioreactor incubates a unique species of the human skin bacteria then. So there's 3 bioreactors. One contains Staphylococcus epidermidis, one contains Corynebacterium, and the other contains Propionibacterium avidum. And I describe these sort of offhand as some being more, in the case of Staphylococcus, smelling more like worker sweat, like a locker room, and others remind me of white collar sweat, the sort of sweat of anxiety and stress. The scents generated in the bioreactors combine in the central chamber, in which a sweatshop icon, the white t-shirt, is infused with their scents, and then they sort of disseminate out with the idea that the scent would intensify throughout the exhibition. the installation was regulated by these controllers that I designed to be more like a factory for continuous rather than batch production. And this for me was biopolitically important in the Foucauldian sense. The control here not just being about laboring bodies, but throughout the entire process of reproduction, growth, death, fully integrated into this industrial process. So the controllers are regulating temperature and pH, heaters and mixers, nitrogen and airflow into the bioreactors, and then nutrient and waste in and out. I described the project in a forthcoming publication as a post-anthropocentric scientific theater in which audiences experience a microbial performance and a simultaneous notable absence. In the middle of the room and at the absolute center of the installation, connected to a radiating network of tubes, cables, and bioreactors, is an empty white t-shirt hanging in a glass bell jar. While humans were once at the center of our worldview, or ontology, or epistemology, in labor, the human has conspicuously vanished. Like Robert Boyle's bell jar at the center of an experimental proof of the existence of a vacuum, and the icon of the emerging scientific episteme of the 17th century, this bell jar rests at the center of a new tentative worldview in which humans have vacated the center, the post-anthropocentric worldview. Along with the installation, I developed this process of making sweat-stained transfer prints in which I could take moist, freshly soiled t-shirts and dust them with charcoal and sandwich them between paper at high pressures, impregnating the paper surface with their image. I first developed the method at Cultivamos Cultura in the São Luís, Portugal, in the summer of 2017. Subsequently, I did these at Microperformativity Symposium in Vienna. What surprised me with these images is how they read almost like medical techniques such as PET scans or fingerprints. It's a bit like fingerprinting in that it makes highly visible marks that are typically difficult to discern. It makes the makes the barely visible visible. So I'll close here with what my expectations for the labor project were, that it would be apprehended differently than other artworks because the mode of perception is primarily olfactory, and that the scents don't correspond to where you expect them to be. And I was hoping that audiences would ponder these perverse contemporary ontologies of production with attributes like life inseparable from labor, product and produce conflated, biology is a subfield of technology, and man omitted from manufacturing. I couldn't have completed the project without the good luck of having the Coalesce Laboratory at the University of Buffalo, which I'm lucky enough to direct. Shown here are some of the different ways that we can cultivate bacteria. This is an anaerobic hood, which can be used to cultivate bacteria that would quickly die in regular oxygen. When I put together these kind of tools for cultivating anaerobic organisms, I assumed that there'd be many such labs on campus, only to find that there weren't, that in many ways, the kind of study of other organisms had been relegated to sort of DNA analysis of these creatures, right? We can, with things like next generation sequencing, we could learn a lot about mysterious organisms with DNA, but little about how they lived or what they ate, or more importantly, how they metabolize or coexist with us. The Coalesce Center is primarily a space that exists as my primary goal of Coalesce has always been to make it a space for sharing knowledge and having residencies. But it was a great place to also make my own work in this case. So the final slide here, and what I'll conclude with, are some of the, I guess, the take-home messages I was hoping to leave you all with. Labor was completed with a different mindset. than most bacterial or bio artworks, and that tradition now it's become common to when one wants to make when one wants to work with bacteria, that one can copy genes from one organism into another and basically genetically engineer them to make the smells that you want. However, in this case, I'm interested in or committed to understanding the organism in all its particulars. How each one of these bacteria live, what kind of conditions they like. For me, the recent understanding of the human microbiome shows that complexity arises through deep organismal processes, and it's not reducible simply to genes. So in conclusion, a few points. One, genetic codes here in this work have little to do with identity or fitness. I want to show that genetic images are not essential. They are cultural constructs, literally built in labs. 3, DNA images are malleable, mutable, fakeable, and their authenticity or their authority can be challenged. And for every technical protocol and standard, there are a myriad of creative inversions that allow audiences to find new meanings. Point 5, our identity can be formed as much by non-humans that affect our characteristics in profound ways and can change based on our environment, our diets, our friends, and our lovers. And while modern science has been preoccupied with differentiating, I'm exploring radical sameness, alikeness, not difference. And I've used in this work a strategy from theorist Bruno Latour, who describes how facts are constructed and authority produced. He describes facts as closed black boxes. He describes the process of closing these boxes as being strong enough to end the controversy, to silence the dissenters. So then to dispute a fact or reopen a closed box requires setting up a counter laboratory to overturn these cultural constructs. So I think at that I will stop my talk and ready yourselves for the next conversation.
Wow, thanks, Paul. That was absolutely amazing. Amazing. Yeah. I'm a bit speechless at everything you just shared. It really, really deeply resonates with many things I think and contemplate. So yeah, can't wait for the talk, which we're gonna be moving into, because it's gonna be fascinating. I think our other two guests are also fascinating, and I think there'll be some really interesting resonances there. So yeah, thank you very much for sharing that. It was
you're very welcome. You're very welcome.
And, yeah, I think we're gonna cut for now, and I'll see you in 5 minutes. Bueno, gracias a todos los que están acá.
Gracias a tú también.
Y bueno, nos vemos en 5 minutos con nuestros otros 2 invitados. Muchas gracias a todos por estar acá en la charla, Paul.